Thank you, Stefan and Dave and the rest of the organizers for putting on a great meeting. I've learned a lot from all the talks and just chatting with people at lunches, so hopefully this will continue, and I'm looking forward to it. Okay, so I'm Albert. I'm from North Carolina State University. It's east coast of the United States. If you ever visit, please drop me a line. Food's not as good as in Paris, but I'll be happy to take you out for a lunch and show you the lab and the campus. A great thing about this meeting is it's one of the first DNA storage focused meetings, and so I can jump right into a lot of the concepts that we've been really interested in for the past almost decade now. So I think a lot of us are here because of the extreme information density of DNA, right? And so that presents a lot of problems, and we've heard a lot of really important talks on DNA synthesis. We'll hear about sequencing, a little bit more about encoding, decoding. What we're interested in is, this is disclaimer, is that if you have extreme information density, that also means that you have extreme molecular and biochemical diversity. So you simply have a lot of different sequences in a small space. And so this, we've heard the density in various formats, 10 to the 19 bases per mil of water. In contrast to the human genome, which is 10 orders of magnitude lower in molecular diversity, this presents both the benefit but also a major challenge, more from a thermodynamic perspective is how we're thinking about it. So if you talk to any molecular or cell biologist that has tried to PCR a specific open reading frame or element out of a genome, the human genome, or even worse, maybe a plant genome, you know that there's a lot of non-specific products that arise, and this occurs at a pretty high frequency. So imagine having to do these types of manipulations at a much higher molecular diversity, where you have much more sequence space that can cause problems. So this will really drive, this theme kind of drives a lot of what I'll talk about today, and has driven a lot of our work. I'll tell you about several different stories. One is one talking about how we can create reusable systems that can be reusable, but a slightly dynamic, a semi-continuous, and that can also compute. We talk about predictive models for DNA binding, and then also an observation we had of non-sequence-based interactions between DNA and how this could be potentially useful for data obfuscation. So I'll start with this first area of creating a reusable store and compute system. This really drives from a lot of conversations at synthetic biology conferences where I might be one of 2 or 3 people interested in data storage, and and there's a lot of skeptics, and so oftentimes people will be like, well, you're using PCR to amplify your files, and then you have to append adapters, and then do NGS, and then analyze the data. It's very batch, low latency, you're destroying the data, it's not reusable. And oftentimes there will be very helpful suggestions of the flavor of, well, why don't you anchor the DNA? So just like a tape drive or a hard disk has positional information, right? Why don't you just anchor the DNA strands in an ordered way, and then you can access the data. The problem with that is that when you go from 3D, a disordered mixture, and you anchor the DNA on a 2D substrate, you give up the vast majority of your information density, right? So that was my initial reaction. You can't do that. You can't anchor the DNA. But, well, and this really leads to this structure that you've seen over many of the presentations so far in this meeting, where you need to use these primers to provide addresses for your files, right? And you're really thinking about your mixture as a highly disordered pool of DNA sequences, and you're basically fishing out what you want using PCR. But we thought about the suggestion of anchoring the DNA and whether this could be useful, and we returned to how biological systems do it, and we thought about this, and in normal cells, DNA is the information template, it's not what you're actually reading in many ways. Instead, you're copying the DNA into RNA, and then you're using the RNA as really the active molecule. And in this case, the DNA is immobilized in a sense. It's not actually physically tethered to anything. It's just itself is immobilized within the nucleus. And it's reusable, and right? Information is repeatedly accessed continuously. Throughout the day, you're constantly transcribing your genes. And despite the fact that it's immobilized, this is what kind of convinced me that maybe we should pursue something along these lines. Despite the fact that the DNA is immobilized within the nucleus, if you calculate all of the DNA within the human body, it still sums up to on the order of zettabytes of data within the volume, right, of your human body. And this is excluding the cytoplasm and interstitial spaces in your tissue and your GI tract, right, which is empty. So you might give up some information density, but you still have quite a bit, right? So maybe it's worth giving this a shot, especially if you think about maybe like a hybrid system where you aren't fully giving up the 3D space advantage, but still anchoring the DNA in some way. So I was talking to a colleague in my department, Orlin Velev, and he had developed these dendrimeric materials with very high surface area. And he had developed these originally for drug delivery and biosensing applications, but we thought, well, can this serve as a scaffold for our DNA? And so we actually did a lot of work where we chemically tried to link DNA covalently to different dendrimeric materials. And we actually found that didn't work very well. It was quite inefficient. The student, Kevin, working on this, he realized, well, you can actually just absorb the DNA to the SCC, just physisorb it, and it'll stick. And you can do this with several different types of polymeric materials, from agarose to cellulose acetate, and you can load DNA onto it in a dose-dependent fashion. These are just some fluorescence micrographs showing using fluorescent DNA showing the absorption of the DNA to the materials. I think I got some of the slides out of order, but what I'm leaving out is that what you can also do is for every strand, you can put a T7 RNA polymerase promoter on it. And so what you can do is you can do in vitro transcription. You can add in RNA polymerase and buffers and reagents, NTPs, and you can flow it through this material and transcribe and then collect the RNA on the other end. And you leave the DNA stuck to this material. So what we found is that this works sometimes and doesn't work in other cases, and it depends on the material that you use. So if you use agarose or cellulose, you see a drop. For agarose especially, which those of you that do DNA gels, it's agarose-based, and it wouldn't be good if DNA was stuck to agarose and wouldn't come off, right? And so that's why you see this big drop. But for cellulose acetate, we actually saw pretty good retention of material, and so we repeated the experiment with just cellulose acetate over 10 rounds of in vitro transcription. We saw that you can actually maintain the RNA yield, and we also checked that you also maintain DNA on the material as well. And so this is a support that allows you to reuse the system over and over again. You can access your data over and over again by transcribing the DNA into RNA without destroying the DNA. You do give up some information density. So I put up some metrics here for properties of the dendrimeric material, as well as what we measured in terms of the amount of DNA that was bound to the material by mass, or if you convert this into terabytes per volume. So this is still pretty good. And so you give up some information density. Like I mentioned, you still retain a lot of that advantage. So there's a little bit of a trade-off, but you still maintain a quite good information density. And so I think a question is this worth it? Are there other features that doing this gets you? So it's reusable, but I think if you're going to look at a data storage system, you're also interested in things like, well, as you repeatedly access the data, what's the fidelity of the data that you get out of that? Robert Grass yesterday talked about, I think it was Robert, talked about how when you do PCRs, and you do multiple PCRs, it's an amplification reaction, and you can get a skew in your population of strands, right? So you can have some strands start to over-dominate your population, and this can cause problems for the amount of sequencing that you need to do in order to recover all of your data. And so what we did was we encoded 3 different files with this type of architecture. So we have a T7 promoter. This is where the RNA polymerase would bind and start transcribing RNA. And then we have some primers here for kind of just downstream processing and quantification studies. And so we used these files to study distributions, changes, skewness in distributions, and recovery of the data and the robustness of those processes. In the interest of time, we'll just jump straight here. What we basically did was we did 10 rounds of data access. So did 10 rounds of in vitro transcription and quantified the abundance of each unique sequence within the files. And what we find is that the distributions don't change considerably over these 10 rounds of IVT. So this is not entirely surprising. I think the key thing that we were worried about was that with each round of IVT, are you potentially losing DNA off the substrate or skewing that in some way? But with this system really being a linear amplification process, and you're always going back to the original template, you're not amplifying and kind of copying, making copies of copies. It makes sense that you would also avoid skewing your, altering your strand distributions in a negative way. We also found that you don't lose any specific sequences. So over these 10 rounds of in vitro transcription, you don't lose any specific sequences, and this allows you to decode the files that you want over and over again. And we also started thinking about, okay, so this system's reusable, but one aspect of reuse would be, well, if you want to use a system where you can potentially store the data for longer periods of time and then rehydrate it and then re-access your data, and you might want to do this a few times, can you do this without degradation to your data? And so freeze thawing is often one of those things that can be detrimental to the stability of your DNA. And so what we did was simply lyophilized the DNA linked to SDCs over 5 different rounds. And then we also did it as a control, just the DNA alone, and lyophilized and rehydrated 5 times. We found that the SDCs was protective of degradation. And again, the distributions were not skewed over 5 rounds. The percent of all unique sequences that were retained remained high. The SDCs, the dendrimeric materials, also seem to have a protective effect through kind of the conventional accelerated aging studies that Robert helped develop several years ago. And so we exposed these SDCs to 65C for 2 days and found that, especially in the lyophilized state, these SDCs protected the DNA from degradation. And if you extrapolate this using the model, models I think several people have used, talked about yesterday, this extrapolates to stabilities that are quite good at both 4C and in frozen state. So this is not entirely unexpected. I think that there's prior evidence that DNA with different types of polymers has some protective effects. So I think that there's a lot of interesting things to look into here from a fundamental perspective. But from a practical one, I think it's nice that you can have a material that in the lyophilized state has quite good protective properties, but could be quickly rehydrated and used in a more warm setting. And just quickly show that in the accelerated aging studies, we also don't see skewness in our distributions. And again, we see maintenance of our all unique strands in our system. Okay, so I've showed you that by immobilizing the DNA on these SDCs, you can you can essentially have a reusable system where you're not destroying the DNA. You can access it over and over again without altering the properties of your files. Another feature of a system that might be a little bit more dynamic, reusable, would be whether you could erase files or data and then reload new data onto it. And so what we did is we had encoded our files with all strands for each file having a restriction enzyme site. And so what we could do is we can add, for example, this should be EcoR1, and that would chew up all of the DNA that has that restriction site in it, and then we can sequence everything else. So if it works pretty well. If we delete file 1, and then we look at the NGS reads, we see no file 1 and only reads from file 2 and file 3. If we delete file 2, it's gone. If we delete file 3, it's gone. And you can decode the other 2 files that are remaining in your system. You can also replace those deleted files with new files. So this is, wherever it's green is just showing that it worked. We could in the NGS, we get the reads that we expect. So for example, here, this is file 1, and you can just erase everything. Just use a DNase that will just chew up all of your DNA, and you can't decode anything because you've erased all the DNA off the SDCs. You can also do things like erase file 1 and then load file 2 onto the system, or load file 3, or you can delete, load, delete, and then load a new file. And you can do this multiple times, and this material is reusable in that sense. And it's also nice that you don't have to chemically link anything. You just flow the DNA in aqueous buffer. And maybe the hallmark of a dynamic system would be, can you compute? And in particular, can you compute in a non-destructive way? So what we looked around and we found some work from Fallhammer and Landweber that was compatible with RNA in particular. So since we're transcribing RNA off of our DNA, we want to use the RNA so we don't have to mess with the original DNA database, right? And so this system was really well tuned for that because you can basically take RNA off of your system. And then the way it works is that you'll have each of these letters represent about a 20-mer sequence. And so, for example, at position A, you can have any number of different 20-mer sequences. Each unique 20-mer sequence could represent something like, in a chessboard, maybe a bishop or a knight. A different sequence would be a rook, for example. You can also abstract this to Sudoku, for example, right, where a sequence, 3 different sequences at the position A can represent a 1, 2, or a 3. And then what you can do is you can add DNA oligos that will hybridize to each of these different sequences and add RNase H, and RNase H will chew up any RNAs that get hybridized by a DNA molecule. So you can, in effect, execute logical operations by adding DNA oligos in specific combinations. And so in order to implement this, well, one of the details I left out is that we've been implementing all of the work that I just talked about within microfluidic channels. You can mix your SDC with magnetic beads and basically immobilize it within a channel and then flow your IVT reagents through it. And what we did was we designed 3 different puzzles, each one having a different RNA polymerase promoter. So in this case, a T7, SP6, or T3 promoter. And if you flow in its corresponding RNA polymerase, you will specifically transcribe that file out. And then you can add specific DNA oligos in different combinations and sequences then to execute your different enzymatic operations. And then all the sequences that you get that are remaining that you sequence through NGS are the solutions to your problem. And so this works pretty well. All of the solutions that we get are correct. And these are just examples. So for example, in the chess problem, what we were asking is, can you spit out all of the board configurations in which this white knight does not attack a black knight? So you see that in the correct solutions, both of these squares are empty because the knight will attack in a kind of an L pattern. And then here are incorrect solutions where a white knight is attacking a black knight. And so the problem in this case was, can you identify all board configurations that in which the white knight does not attack a black knight. So this worked pretty well. Okay, so just to summarize, kind of give you an overall picture of what the system looks like, we immobilize DNA onto these high surface area dendrocolloids. They maintain pretty high information density at 10,000 terabytes per cubic centimeter. They're protective of the DNA. You can transcribe the DNA into RNA in a semi-continuous fashion. You can erase and reload files. I didn't talk about this, but you can also just directly sequence the RNA using Oxford Nanopore. And then you can compute with the RNA, all of this being non-destructive to the original DNA that's in your system. So stay back here a little bit. Okay, so I think one of the messages I hope to communicate outside of this community and maybe to skeptics is that we've focused, I think, a lot about talking about how DNA storage really focuses on archival. And I think in the lay public and maybe scientists outside of the realm of DNA storage, a lot of them are caught up over this fact that, well, you can't create anything that's remotely similar to electronic systems, right? But I think we can make the argument that we just don't know. And it's possible that you can have systems that can bridge this gap. And from a practical perspective, I think it could also be useful, right? That you can have archival storage that's easily rehydrated and activated to move into these kind of short-term dynamic settings. And I think this material and immobilizing DNA onto materials can be one path to get at that. And it fulfills a lot of the requirements that we want. It helps stabilize the DNA, but also helps us perform dynamic processes. So I'll return also to this analogy to cells, right, that I talked about at the beginning, about how cells immobilize DNA and it could be a good way to store data. I originally, I think, avoided the idea of storing data within cells simply because the delivery of DNA into the cells seemed challenging. But I think we're early on, and so we should explore a lot of different ideas. And cells do have a couple advantages. One is that it replicates the DNA for you. If it's a dividing a mitotic system, and it also helps preserve the DNA. We also wanted to think about this other question of this high molecular diversity, this high biochemical diversity. And for example, like how do you create addresses for your system, right? And in biological systems, a lot of molecular recognition is not necessarily just based on DNA hybridization, actually very little of it, maybe like microRNAs or systems like that. But in fact, a lot of the molecular recognition in natural systems is actually done with proteins. And there's probably a lot of reasons for this. Maybe the top two is that for DNA, your interactions are really governed at a 1D to maybe 2D level, if you're thinking about hairpinning. For proteins, you're really taking advantage of 3D shape. And I put 4D here because I think there's a lot of evidence also that kind of dynamic breathing and conformational changes in proteins is important for binding and protein function as well. You're also moving from ACTG to 20 amino acids. So you have a larger chemical space to work with as well. So we did a very simple we created a very simple system using something called yeast surface display. So in this scheme, you take DNA oligos that encode different files, and you can introduce them into the yeast genome. And the yeast can also use the system that it naturally has to display different proteins or peptides on its surface. So this is tethered to the surface, but it's encoded by DNA you introduce into the cell. So this is distinct from the DNA that's encoding your file. It's rather just a peptide sequence that you're using as an address. So instead of a 20-mer primer sequence, you're using a peptide or a protein sequence as the address. And so our hope is that if you use proteins, you can really expand the address database that you have possible. And so what you can do is then you can use magnetic beads that have something that binds each of these different addresses uniquely. Now, one of the advantages I talked about using cells is that they can replicate, and one of the, I think an open question is, as cells replicate, again, do you skew the distribution of files that you have in your system? So we checked for this, and over at least 20 cell divisions, you actually maintain the strand abundance pretty well. I think this is slightly better than PCR, so if you were to do 20 cycles of PCR, for example, you would actually get a considerable skew in your strand distributions of your files. You also maintain a similar strand retention, and it doesn't seem to introduce many errors. So the blue would be your initial library that you order from Twist or GenScript. And over 20 cell divisions, we didn't see an increase in the error rate of your system. So it does seem like storing information in cells have some benefit from those perspectives. And we also showed that you can access the file using these protein-based addresses. So if you access just file 2 using the epitope that's displayed on file 2 yeast, you can extract file 2 sequences, and similarly, you can extract file 3 using a different epitope. I also note here, you can start to do some weird, more complicated file accesses or random accesses by using proteins and peptide pairings that are not simply one-to-one, but rather have 1-to-2 or overlapping types of interactions. So for example, there are these chromatin peptides, histones with lysines that are methylated, that are known to bind several different proteins within the human genome. And so you can use these peptides to label, for example, file 2 and file 3 yeast, and extract both files at the same time using a single protein. So, and using different peptides, you can extract different combinations, for example, file 1 and 3 or all 3. So this leads to the possibility of creating kind of database organization and file access systems that are not just simple one-to-one pairings, not simply orthogonal to each other, but you actually create more complicated networks and that can support things like maybe search. So this really kind of transitions us to thinking about whether we can create predictive models of biomolecular interactions. I'm going to switch back to DNA now because I think the protein world is a little bit hard to do this predictive work, although there's a lot of advances here. Also, I think a lot of us are still interested in DNA and as ways to organize and address data. So I think what really drives understanding how DNA interactions are governed, it really comes down to thermodynamics, right? And the problem, I think, can be illustrated by the simple diagram where you have delta G, you have a specific DNA sequence, and it will bind its perfect complement, but we also know that it will bind other sequences that are similar. They might have a few mismatches here and there, but they will still bind. And we can quantify this experimentally, as well as through simulations, where we take a sequence and then measure its binding to other sequences with increasing number of mismatches as you go from left to right. And the y-axis is showing the percent of these pairings that bind. And what we can see is that even up to 5 and even up to 10 numbers of mismatches, in this case Hamming distance, you can actually still get productive binding. So what this means is that if you want to create orthogonal addresses, so PCR binding sites, you have to basically design them to all be about maybe 7 or 8 Hamming distances apart. What this means is you take a 4 to the 20 potential space and reduce it to something that's more like 30,000. So it's quite limiting because of this issue of nonspecific interactions. This problem is not just to do with the primer addresses for PCR, but you also have to avoid PCR primers binding to the middle the payload regions of your DNA sequences. So in PCR, you have two strands and they come apart, right? In every cycle you're melting them and then and then you drop the temperature and the primers will bind. And so they have the opportunity to not just bind the ends, where the primer addresses, but they can also bind in the middle of your sequences. So what this means is that you also have to design your encoding so that the payload regions do not clash with your primers. That's quite restrictive. That's basically illustrated here. The key idea here is basically that you get capped at the database capacity that you can operate at simply because you have to, as your system has more and more addresses, more and more files, it gets more and more restrictive for what the sequences can be in your data payload. You have to encode it in a less dense and less dense fashion. And so you get this cap. So I'll tell you about one quick thing that we did to address this, one really simple thing, which is just create a system where you have a hybrid structure, where you have a double-stranded region, so your data payload's blocked. And then you have a single-stranded region that at room temperature, you can have something bind it, and then you can separate it using a magnetic bead, for example. And so you avoid this problem of this primer or this oligo binding to the middle of your payload. This frees you up to basically encode your payload region with whatever type of encoding that you want. And this allows you to scale your database capacity without restriction. We also thought about, let's come back to the file addresses in the more conventional setting. Can we take advantage of the fact that you get binding to non-specific sequences? And we borrow some basic knowledge from just when you run PCRs that temperature and concentration matter, and so you can actually tune these interactions. So for example, if you increase the primer concentration, you can make PCRs much more promiscuous. You can start to have productive PCRs with things that even have 15 Hamming distance difference. That's represented here by basically this primer being able to amplify sequences within this gray sphere and interacting and amplifying with sequences that are of increasing Hamming distance away from it. You can also make the interactions more stringent by increasing temperature. That's shown here. You can shift this curve to the left and make this sphere of interaction smaller. So why could this be useful? We thought about things like file preview. If you have a Mac, you press spacebar, you see a kind of a lower resolution image of your file. So the idea here is that we encoded low resolution black, white, or grayscale data with perfect addresses to the primers that we're using to access the data. And then everything else, the higher resolution data, color information, we encode with addresses that have 4 or 5 Hamming distance difference from the PCR primers. And so in a stringent condition, we access only this kind of low resolution, saves on sequencing costs. You don't have to sequence as much. You can kind of check, do you have what you want? And then if you want to access all of the data, then you switch to a lower temperature, higher primer concentration, and run the same PCR, and you can access the full data. And this is the NGS raw data here. So this idea kind of led us to think, well, instead of just thinking about trying to create orthogonal interactions, is there utility? I think there is utility to working within this kind of off-target binding space, a parameter space. So the preview idea is one, but also work from UW and Microsoft thinking about search by using non-perfect interactions between DNA. And in order to be able to do this type of work and implement search, preview, other functionalities that we might not have even thought of yet, what we really want to do is be able to predict, right? We want to create models that can predict these off-target interactions. Right now, in those two examples I just gave you, we did this kind of phenomenologically, empirically. We found things that seemed to bind off-target and used those. can we actually design and predict and engineer these non-specific reactions? Currently, our systems look something like this, where this kind of inner circle of nodes or DNA sequences interact with this kind of outer circle in this kind of graph network, but they don't interact with any kind of overlapping nodes, right? So this orange one interacts with these two nodes, but it doesn't interact with any of the nodes that this blue sequence interacts with. So that's kind of where we're trying to engineer things orthogonally, but what we really want to do is create something that's more complicated. And so an example I use here is hyperconnected networks. I'm a millennial, so I know a lot about memes, and I realize that a lot of people don't do memes anymore. But basically, the idea of a meme is that you can have the same data, but then put different labels on them, right? Or in this case, you have the same text, but different images. So if you were to store this data, would you store each 4 of these separately, or could you be more efficient and somehow store this data and this data once, but be able to access it in different combinations? So this is basically, can you think about deduplicating data? Can you move from a system that looks something like this to something that looks like this, where you start to have DNA-DNA interactions that they have DNA sequences interact with multiple other DNA sequences, and can you actually control this network and predict it and engineer it? So this is a really hard problem because already, just creating orthogonal sequences is hard, but can you create these type of overlapping networks? That becomes very, very tricky. And I think what's really holding us back from creating these kind of predictive models is the sparsity of wet lab data. If you look at what's been used to train NUPAC, Primer 3, and others, oftentimes they're actually based on perfect sequences with other sequences that are maybe a few mutations away from it. What we want to think about are things that are 5, 10 Hamming distances or edit distances away. So one problem is data sparsity. And the other problem is simply the large parameter space that you have to be able to predict from. So I'm going to tell you about how we address this and how we're hoping to kind of tackle that in the coming years. So to just generate data, what we did was created a DNA architecture that looks like this, where you have a double-stranded DNA region, and this provides an area where we can just put barcodes. This allows us to do sequencing in a multiplex fashion. And then we have a single-stranded DNA overhang region. This is completely random. This is where you add all 4 nucleotides in every round of phosphoramidite chemistry. So this basically represents our prey library, what I'll call the prey library. And then we designed 26 different baits that are biotinylated. So these are single-stranded DNAs. They're 20 in length. They're biotinylated. We mix these and then pull out everything that binds to the sequence. So we're essentially panning about 1 trillion sequences out of here. And then we take what's bound, we also take what's unbound, and then we sequenced in total about 100 million reads from all of that. So 100 million is actually a really small percentage. It's like less than 0.01% of this possible sequence space, but it's still really expensive to sequence. And so before we did that, we wanted to make sure that there was some metric that gave us confidence that this was working. And so what we would expect is that most of these random sequences would not bind a fixed 20-mer sequence, right? And so we would expect that the pullout would give you a very, very small yield in terms of DNA mass. And the way we did this was that we had enough binding sites so that theoretically every single molecule from this library had the opportunity to be bound and pulled out. So when we quantify the amount of DNA that's actually pulled out, we see that it's quite low relative to the unbound, and so this was what we expected. And so we went ahead with the sequencing. We did the 100 million sequencing reads. And we found that also if you looked at each bait primer, so we did 26 different pullouts, and each pullout we saw that there was very little overlap in shared sequences that was being pulled out. And this was just giving us confirmation that we weren't having some weird non-specific effect that was occurring. So the next thing we did was to see whether enrichment was actually occurring or whether we were just getting non-specific noise in the experiment. And so what we used was edit distance as well as delta G to plot all of our reads from the bound population in green as well as the unbound populations in orange and the original library in blue. And you can see that as expected, you get a shift in all the bound sequences. They all have lower delta Gs and lower edit distances, as you might expect. But I think this is also illustrative that these metrics aren't great, right? If you're using these to design DNA-DNA binding, this is not a great metric because you have substantial overlap between your bound and unbound populations. From here to here, basically, it's a bit of a toss-up as to whether you would predict these things to bound or not. And so what we did was we fed all this data into a CNN. This is a confusion matrix here. I think I'm going a little slow today, so I'll speed up, but basically, this is the ROC. We're getting accuracies roughly over 80%. I think what's more useful to see is plotting the model's prediction probability for all of the different sequences, similar to how I showed you edit distance delta G. And what you see is that the bound and unbound populations bifurcate pretty strongly now. They don't really overlap like they did before. If you do this for all 26 data sets, so each row is one of the 26 data sets, you again see that you get this bifurcation between the bound and unbound population. Whereas if you look at edit distance and delta G based approaches, you still maintain a lot of overlap between your bound and your unbound data sets. So this CNN is allowing us to have much more discrimination in our predictions. The system has just over 80% accuracy. It does better head-to-head against NUPAC primer 3 delta G based models as well as edit distance. It runs faster. I also want to point out that what's really interesting is that when we first did this, we were constrained by cost, so we did 26 different bait primers, bait oligos. We thought that the more we did, the model will continue to improve in accuracy. But what we actually found is that if you train on one or two and then test on all the others and continue to increase the number of data sets that you train on, you actually can reach this plateau pretty quickly. So this suggests that our model is demonstrating pretty data-efficient learning and generalizing well from limited data, and that perhaps the space of DNA-DNA interactions and how we think about this is not as complicated as we might think. Okay, I'll skip over this, but just note that this. Okay, I'll talk about this. So we're really thinking about hyperconnected networks. So what we wanted to do is take this model and think about whether we can actually create a hyperconnected network. Something looks like this, where you have 26 bait sequences represented by this inner ring of nodes, and then 96 prey sequences represented by this outer ring. And what this is showing is that each node can interact with many other prey sequences, but not in a one-to-one fashion. You're actually having each one interact with a complex subset of preys. We designed the baits using edit distance to make sure that each of these 26 were pretty far apart from each other, pretty dissimilar. And then this is another way to represent this network here. So each row represents one bait sequence, and each column represents a prey sequence. And the yellow is where we want a bait to bind a prey. So this looks a little, I don't think this is super intuitive, so I just want to highlight this row, for example, this top row. It won't bind, we don't want it to bind this prey sequence, but then we want it to bind these next three prey sequences. The second bait, we don't want it to bind these two, but then we want it to bind the next three. So we wanted to bind we wanted to bind some of the same sequences, but not all of them. And we want to do that for this entire pattern here. This is actually pretty challenging to do, but it works to a certain extent. So everywhere you see green is good, everywhere you see red and pink is bad. And so our model does pretty well. If you compare that to NUPAC, NUPAC pretty much was not able to do this at all. the accuracy does drop considerably, but is still, but does a lot better than prior systems. So this is, I think, a pretty hard problem to create, and so we're trying to continuously improve this model so that we can boost this accuracy further. Okay, so I told you about some predictive models. I think, I think in the interest of time, I might, 5 minutes? Okay. Okay, I think I have enough time. I'll tell you about something that's a little bit different. So everything I've been telling you up to here is kind of, we're trying to design something, right? We're trying to make certain DNA-DNA interactions occur. We're trying to design it. One of our students, Sneha, she was just like, well, one day bored and just threw some DNA in a dynamic light scattering apparatus. And she was expecting, so this is just a PCR product, and she was expecting to see a single peak at this size, but instead she saw these small peaks at a higher size. And initially, she was collaborating with another student. She was like, kind of teased the other student, you gave me kind of dirty DNA. But we realized that it was pure DNA, and what happened was that she used PBS, which has a little bit of an ionic strength. And so she titrated the salt concentration higher and higher and found that you would shift from this lower peak at low salt to this higher peak at high salt. And we thought, okay, maybe the DNA is aggregating, but didn't really believe it until we looked at TEM, and this is for single-stranded and double-stranded, and you can see they form different types of nanoclusters, I think probably because the single-stranded are hybridizing to a certain extent, whereas the DNA is clustering not in a hybridization-based manner, but in a more nonspecific backbone-mediated manner. Okay, so it turns out that this is also reversible. So you could start at room temperature and start to heat it up to 60 C, which is actually still well below the hybridization melting temperature. And when you reach the 60 C, you can break apart the aggregates. And then if you cool it back down, it returns back down to the smaller, to the larger aggregated size. This is also reversible with mechanical shear. So you can start with a nanoclustered population, just put it on a vortex, break apart the clusters, and then leave it undisturbed for a little bit, and it'll reform these aggregates. If you use really high ionic strength, you prevent this breakage from occurring. So we thought about how this could be useful, and what we did was took two different files that are comprised of about 1,000 distinct DNA strands each, and we're thinking about, okay, we just don't want to care about addresses, so we're not accessing these files using PCR or anything like that. Instead, what we're going to do is take these strands and mix it with a constant S1 sequence for file 1 and cluster those together. And then we're going to take file 2's sequences, cluster those with a different DNA molecule S2, different sequence, cluster those together. And then the idea would be if you mix these two clusters together and then use an oligo to pull out against just S2, you should also pull out all of the strands from file 2. And if they're not breaking apart and not intermixing, then you wouldn't get anything from file 1. And so that's what we saw. You mix these two aggregates together, you mix them, and then you use an oligo to pull out just S2, and you get only strands from file 2. If you vortex this mixture, you break apart these nanoaggregates, you let it reform, and then they now reform in a scrambled manner. Now S1 and S2 are all scrambled with file 1 and file 2. You cannot distinguish these nanoaggregates apart from each other. When you go in and try to extract file 2 alone, just using the S2 binding sequence, you get sequences from both files. So this is an example of essentially obfuscating the data or scrambling the data from simply just vortexing these mixtures. So one way you can think about it is these nanoaggregates are essentially kinetically trapped aggregates. Okay, so just kind of return back to end by kind of summarizing our we've really been thinking a lot about how do you deal with this problem of really complex biochemically and molecularly diverse mixtures. This poses a lot of interesting problems, but also advantages and new functionalities you can derive out of them. I told you about a reusable store and compute system, a machine learned predictive model of DNA dating binding that I think will be useful for not only creating orthogonal primer sets, but also designing interactions that are off-target, so-called off-target, and then also that there's a lot of interesting biophysical properties of these complex systems that I think that are kind of still up to be explored and leveraged. So with that, I want to thank students in my group that have done all of the work. Our collaborator, James Tuck, who's a longtime computational theoretical collaborator, and his students. Orlin Velov developed the soft engineered colloidal materials. Winston Tim helped us with ONT reading of direct ONT readings of RNA, and Adriana San Miguel helped us with implementing that store and compute engine in a microfluidic setup. Okay, with that, thank you for your time. Thanks for getting up for the first talk of the day. Happy to take questions.
Any questions?
Thank you for the talk. So when we're talking about the extreme density of DNA, quite often we think we're going to have a very small number of copies for each one of the strands. And I was wondering if, so you talked about the delta G, if you had studied also the kinetics of strands actually meeting when they're in very dilute conditions.
Yeah, so this is a great question. It's like, so I'm a chemical engineer by training, and for the first 5 years of my lab, I felt a little bit like a fraud of chemie because I didn't do that much chemie. But then we started thinking about this, then we started thinking about this from like a chemical kinetics and thermodynamics perspective. I think what you're asking is, what's the time scale of a molecule, like one DNA finding all of the DNAs it needs to find, right? Yeah, so it's really interesting. If you were to think about just diffusion, so there's basically two types of transport that molecules can take. So there's a diffusive transport. If you rely just on diffusion, so just a molecule moving in a fluid, it takes something crazy like 3 hours for a molecule to move like a micrometer. You won't believe it, but if you look at the diffusion coefficients for the molecules of this size, it's quite limited. Actually, what you're getting, why mixing, if you just add DNA to a solution, is because you have a lot of convective transport as well. That's due to temperature gradients as well as just mechanical mixing. At the scale of like a mill, it's pretty good. If you start to increase your volumes beyond a mill, and you increase the molecular diversity of your system, you do run into problems where the timescales of a molecule sampling all other molecules will be like millennia. And so we have to think about how many input strands that we're putting in order to be able to sample everything, balancing the concentrations of the input relative to what's in the database, as well as the sheer scale. And also probably we have to think about the viscosity of the system, because that also affects the convective and diffusive transports. But I think that's kind of like understudied or underthought of problem. So I think you're very astute to intuit this as an issue. I think the key thing to remember is that a molecule moving a millimeter in fluid just by diffusion is like days to do that. Yeah.
Thank you so much for sharing your work. You have a lot going on, I think. I had a question about some of your earlier slides. You had the diagram where you have the dendritic colloids, and the magnet is like immobilizing them. I might have missed, how are you attaching magnetic beads to the colloids or to the DNA? How are you trapping them with a magnet?
Yeah, that's exactly right. We found that just like you can absorb DNA onto this material, it's pretty sticky to magnetic beads too. So you can just mix in some magnetic particles, and even iron oxide would work, and they'll just tangle in with your SDC, and then you can just use a magnet to immobilize it. Yeah.
Thank you. Very beautiful talk. A similar question on this topic. So you mentioned the DNA is just adsorbed on the matrix, but still it's accessible to enzymatic reactions. I mean, the RNA polymerase has to open the duplex, get in. How do you think that it's possible that the adsorption is still stable, but the access is still possible for the enzyme?
It's a great question. So I think about it like a little bit like how in the human or eukaryotic genomes, DNA is wound around nucleosome protein complexes, and as the DNA polymerizes around it, it doesn't kick the nucleosome off. Rather, it walks across the nucleosome. I do think if you're, and so the DNA is probably staying bound largely to the polymeric material throughout the transcription process. What we did find that kind of supports this theory is that if you shorten the template to a level that's like maybe 70 or 80 nucleotides or base pairs long, you lose the DNA. The DNA starts to come off, yeah. So you have to have a long enough piece of DNA to stay stably on there.
Thank you very much for your talk. I'm new to this field, but as a biologist, I was wondering, how do you assess, when you're talking about storage within the cell, using histone modification to retrieve your sequence, how do you decide, how do you make, or how do you label a specific file with a specific histone modification?
Yeah, thanks. So in this case, what we're doing is we have chromodomains or bromodomain proteins. So these are classes of proteins that will bind methylated or acetylated histone peptides. And those are the proteins that we display on the surface. So each file, each yeast population that contains one file will have like bromodomain 1. And this one might have bromodomain 2, and so on. And then what we do is we have a magnetic bead that has the peptide, which is chemically synthesizable. So it's easier than putting the protein on there. Instead, we have the peptide on the magnetic bead. And then those have different modification patterns that we can put on there. And then we use those to pan out specific binding partners. So that's also why, if you have two bromodomains that are slightly different, they can often bind the same acetylated peptides. And then there are some that they bind distinctly. So you can get those kind of interesting overlapping patterns of access. Yeah.
Thank you.
Any other question? I have one for you, Albert. Using transcription is mimicking our cells compute and how they store data. Do you think what you've done is scalable? I mean, in vitro transcription is extremely expensive, very complex to control. What you showed for deleting some files using one specific restriction enzyme or expressing from one single promoter, seems to me that you don't have enough tools to make all this, and rather it would be, if you want to use transcription, I would rather use it in a cell rather than in vitro. What is your point of view on this?
Yeah. So it's packed with a lot of stuff. So one of the things that got us started in the field was when we wanted to PCR a small file out of a large database. So we wanted to do something where we PCR'd a kilobyte file out of like a gigabyte or terabyte background of data. And what we found is that you can't do that, because even when you amplify that file enough, like 30 cycles or even 60 cycles, you run out of DNTPs to actually overwhelm the rest of the data that's in your system. And so one of the things that I think is useful about this type of approach, and it's not specific to transcription actually, is just by anchoring your data and then flowing what you access out, you remove all that background noise. So that's one of the principles that is not necessarily in vitro transcription related, but I think it's useful. PCR versus IVT, I think cost-wise, it might not be that different. It's NTPs and transcriptase versus DNTPs and polymerase, and it's just, from an industrial perspective, which one are you scaling up, and you'll optimize the processes to make those. I don't think there's anything too inherently different from a cost perspective. I think this question of scaling the file access and deletion, that's tricky, right? And that's kind of a similar problem, I think, with DNA. How do you address things in a specific way? I showed things using restriction enzymes for deleting files, using a few different polymerase promoters for accessing files. That's not scalable, right? Like, there's not enough restriction enzymes out there. But you could imagine maybe using CRISPR-based systems where you have 20 more sequences that you're targeting to delete a file using a sequence-specific endonuclease like CRISPR. You could even use CRISPR to target polymerases and avoid using, so basically diversify the types of promoters that you can use. So there's potential tricks like that you could take as well.
Okay, thank you very much.